हेलो बच्चों, वेलकम टू SPSVERSITY. In the previous session, we discussed about biomolecules, in which we discussed about biomicromolecules, macromolecules, enzymes functioning, and their different types of enzymes. So in this session, we are going to discuss about cell cycle and cell division. So what is this cell cycle? So cell cycle is a sequence of events. So we can see events which occur in sequence where the DNA gets replicated and synthesize other constituents of the cell. Other constituents means like proteins, cell organelles, and ultimately form into two daughter cells. So इसको क्या बोलते हैं कि cell cycle. So it is a sequence of events which occur in a regular manner. And finally, we'll get two daughter cells from one parent cell. So cell cycle में एक parent cell से दो daughter cells आता है. और दो daughter cells, they are similar to parent cells. और daughter cells में chromosome number और पेरेंट सेल में क्रोमोसोम नंबर सेम रहता. अभी आप क्या देख रहे पिक्चर है कि द टोटल इवेंट्स ऑफ सेल साइकिल, इसमें आप देख सकते इंटरफेस. Next phase is M phase, metaphasic plate. So interphase और mitotic phase. So cell cycle में दो phases रहते और interphase में 3 sub phases रहते. So G1, S, G2. So mitosis में prophase, metaphase, anaphase, telophase और cytokinesis रहता. This is, it shows the sequence of events in a proper way. Which events occur after which event. Okay. So if you take the phases of cell cycle, cell cycle में majorly we can divide into 3 phases: interphase, mitosis, and cytokinesis. So interphase में 3 phases रहता. Is G1, which is G means gap, S means synthetic phase, where DNA synthesis takes place. So S का full form क्या है कि synthetic phase. Okay. G2 gap 2 phase. So interphase में 3 phases G1, S और G2 phase. Mitosis में 4 phases रहते, इसमें 4 phases हैं. So पहला phase prophase, next metaphase, anaphase और finally telophase. साइटोकाइनेसिस में क्या होता है कि डिवीजन ऑफ साइटोप्लाज्म. माइटोसिस में division of nucleus होता है. Nucleus will divide into one nucleus will divide into two nuclei. एक nucleus से दो nuclei आता है. Okay. और इंटरफेस में कोई डिवीजन ऑफ न्यूक्लियस नहीं रहता और डिवीजन ऑफ साइटोप्लाज्म नहीं रहता. इसलिए इसको क्या बोलते कि पूरा इंटरफेस को क्या बोलते कि रेस्टिंग फेज. ओनली बायोकेमिकल चेंजेस आर सीन इन दिस. इसमें सिर्फ बायोकेमिकल चेंजेस होता. नो डिवीजन ऑफ न्यूक्लियस, नो डिवीजन ऑफ साइटोप्लाज्म. सिर्फ माइटोसिस में डिवीजन ऑफ न्यूक्लियस होता. साइटोकाइनेसिस में डिवीजन ऑफ साइटोप्लाज्म. वे हमेंगे इंटरफेस. इंटरफेस को क्या बोलते कि रेस्टिंग फेज. सिर्फ इसमें ओनली बायोकेमिकल चेंजेस रहता. इसमें 3 स्टेजेस रहता. फर्स्ट ग्रोथ फेज, that is G1 phase, gap 1. S phase, that is where DNA replication takes place, DNA synthesis phase. Second growth phase, इसको क्या बोलते हैं कि gap 2 phase. So G1, S phase, G2 phase. So इसमें 3 phases रहते. पहले मैं G1 फेज के बारे में. G1 फर्स्ट गैप फेज, दैट इज ग्रोथ फेज 1. इसमें ऑर्गेनेल्स आर प्रोड्यूस्ड. दैट मीन्स नथिंग बट सेल ऑर्गेनेल्स आर प्रोड्यूस्ड. और साइटोप्लाज्म का वॉल्यूम इंक्रीज होता है. So size of the cell also will increase. Volume of cytoplasm will increase. So in G1 phase में ये changes होता. Synthesis of organelles, increase in volume of cytoplasm. So पहले तो. So first gap phase where you can see the synthesis of mRNA. So basically for synthesis of proteins पहले mRNA चाहिए, बाद में mRNA से प्रोटीन्स आता है. And next phase is synthetic phase. इसमें DNA replication. Here DNA replication means इसमें DNA number becomes double. So DNA becomes double, but chromosome number remains the same. Chromosome number will not change. Only DNA count becomes double. If it is 2C, the DNA count, after synthetic phase, it will become 4C. Just doubling of DNA takes place. Next phase है कि second gap phase. इसमें क्या होता? इसमें microtubules are synthesized, which are nothing but which are made up of tubulin protein, which are required for movement of chromosomes during mitosis. So mitosis में chromosomes का movement रहता. इसके लिए microtubules चाहिए. So these microtubules are synthesized in second gap phase, which is nothing but G2 phase. This is what happens in second gap phase. Okay, so here you can see in G2 phase, okay, microtubules are synthesized. So next we'll come to mitotic phase. So mitotic phase means from one nucleus, two nuclei are formed. And each nucleus will have the same chromosome number as the original cell is having. If the original cell is having 46 chromosomes, the daughter nuclei will also have 46 chromosomes. Same chromosome number. There is no change in chromosome number. और mitotic phase में 4 phases रहता, prophase, metaphase, anaphase और telophase. ये phases को इस सीक्वेंस में रिमेंबर करना है, याद रखना है. इंटरफेज में भी G1, S, G2 phase, the same sequence you should remember. In mitotic phase, prophase, metaphase, anaphase, telophase, you should remember in the same sequence. Okay. So after interphase, the cell enters into mitotic phase. M phase, in short form, it is called M phase. A M phase में पहला phase prophase, next metaphase, anaphase, और last finally it is telophase. 4 phases. So पहले हम आएंगे prophase के बारे में. Pro means the first phase. So prophase में क्या होता है कि chromatin material will condense and form chromosomes. So chromatin material means which is thread-like. The DNA is present in the form of a thread-like. इसको क्या बोलते हैं कि chromatin material. So this chromatin material will form into a rod-shaped structures. So इसको क्या बोलते हैं कि chromosomes. इसको क्या बोलते हैं chromosomes. So chromatin material से chromosomes form. After formation of chromosomes, the nuclear membrane disappear, or nucleolus also disappears. So in prophase, the nuclear membrane, the nucleolus will disappear. Chromosomes form. So what you are seeing in this picture, this is chromatin material. A chromatin material condensation के बाद में chromosomes form होता है. So it will form chromosomes. Next phase की metaphase. Meta means middle. Meta means middle. Metaphase में क्या होता है कि spindle fibers are attached to kinetochores of chromosomes. So chromosomes में chromosomes का parts में लिया तो the center one, इसको क्या बोलते हैं कि centromere. Center part को क्या बोलते हैं कि centromere. A centromere में on either sides kinetochores रहता, proteins रहता, उसको क्या बोलते हैं kinetochores. Spindle fibers are attached to these kinetochores. After attachment of these spindle fibers, these chromosomes are moved to equator. Equator means the middle of the cell. Equator means the middle of the cell. And all the chromosomes are aligned in the middle of the cell like a plate. इसको क्या बोलते हैं कि metaphasic plate. इसको क्या बोलते हैं कि metaphasic plate. A metaphase में पूरा chromosomes center of the cell में रहता with the help of spindle fibers. So with the help of spindle fibers. So here, if you look into picture of the metaphase, so metaphase में chromosomes center of the cell में इसको क्या बोलते हैं इक्वेटर ये पूरा chromosomes को मेटाफेज spindle fibers are attached to the kinetochores of the chromosomes. So इसको क्या बोलते हैं कि poles इसको poles बोलते हैं. A spindle fiber, this entire structure of spindle fibers, it is called spindle apparatus. इसको क्या बोलते हैं कि पूरा spindle apparatus. A spindle apparatus में और इसमें सब types रहते spindle fibers. Continuous, discontinuous spindle fibers. Okay. So like this, with the help of spindle fibers, the chromosomes are aligned in the center of the cell. इसको क्या बोलते हैं कि मेटाफेज प्लेट, ए पोल्स. ए सेंटर वन इसको क्या बोलते हैं कि सेंट्रोमियर. सो नेक्स्ट डिवीजन क्या आता है कि एनाफेज. एनाफेज में क्या होता है? एनाफेज में सेंट्रोमियर विल स्प्लिट. सो स्प्लिटिंग ऑफ सेंट्रोमियर्स. सो स्प्लिटिंग ऑफ सेंट्रोमियर के वास्ते क्या होता है कि क्रोमैटिड्स और सेपरेटेड. That is, if you take the chromosome, a centromere, a chromatids. Due to splitting of centromere, chromatids separated. And these chromatids will move to opposite poles. A ऐसा डायरेक्शन में होता, A ऐसा डायरेक्शन में जाता. So chromatids move in opposite poles. So एनाफेज में changes क्या होता है कि splitting of centromere, where after splitting of centromere, chromatids moves to opposite poles. Okay. After anaphase, chromosomes will form at each pole a cluster. Okay. After anaphase, the next change phase is telophase. A telophase में देख सकते हैं group of chromosomes. Cluster means group. At each pole, group of chromosomes. Okay. बाद में क्या होता है कि nuclear envelope is formed. Again, nuclear envelope is formed. Nucleolus is formed. Golgi complex or endoplasmic reticulum is there again. So telophase is quite opposite to prophase. The changes which occur in telophase are quite opposite to prophase. Okay. Prophase में from chromatin material chromosomes are formed. इधर क्या होता है कि chromosomes से chromatin material. Prophase में nuclear envelope, nucleolus disappears. Telophase में nuclear envelope और nucleolus are again reformed. So इसको क्या बोलते हैं कि telophase. A telophase से mitosis phase is completed. Okay. So mitosis phase is complete. After completion of mitosis, the next is division of cytoplasm. That is cytokinesis. A cytokinesis में animals में different process रहता और plant cells में different process. Why it is different? Why? Because in animal cells there is no cell wall. Plant cells में cell wall. थोड़ा differences रहता cytokinesis और cytokinesis in plant cell और animal cell में थोड़ा difference होता. So plant cells में cell wall रहता, cell wall formation starts from the center. इसलिए different cell plate method, इसको क्या बोलते हैं कि cell plate method. Animal cells में cell wall नहीं रहता, सिर्फ plasma membrane रहता. Just division of the formation of plasma membrane. It is furrow method. Either it is by cell plate method or furrow method, finally cytoplasm will divide. So after division of cytoplasm, so two nuclei, that is two cells are formed from one original cell. एक सेल से दो cells form होते. ऐसे हमें डिस्कस किया साइटोकाइनेसिस में प्लांट्स में थोड़ा अलग प्रोसेस रहता है, एनिमल्स में थोड़ा अलग प्रोसेस रहता है. ऐसा हम देख सकते हैं हियर प्लांट एंड एनिमल सेल्स इन प्रोफेज सिमिलर रहता, जस्ट अ फ्यू डिफरेंसेस लाइक न्यूक्लियोलस डिसअपियर्स, न्यूक्लियर मेम्ब्रेन डिसअपियर्स. Chromatin material will condense and form chromosomes. Animals में आए तो centrosomes are formed, that is separation of centrosomes. Centrosomes से plants में नहीं रहता, सिर्फ animals में cell में रहता, सिर्फ animal cells में रहता. Okay. A mitotic spindle which is formed. Metaphase, no changes between plant and animal cells. Chromosomes are aligned in the center of metaphasic plate. A picture में देख सकते, these are spindle fibers. These are chromosomes to which spindle fibers are attached to kinetochores of centromere. Only they are attached to kinetochore, nowhere they are attached to other parts. ये पूरा स्ट्रक्चर को क्या बोलते हैं कि मेटाफेज प्लेट. इट इज इक्वेटर, द सेंटर ऑफ द सेल इज इक्वेटर, द सेंटर ऑफ द सेल इज इक्वेटर. एनाफेज में क्या होता है कि स्प्लिटिंग ऑफ सेंट्रोमियर होता है. Due to splitting of centromere, chromatids separate and move towards poles. Okay. And these unattached microtubules push against each other to elongate cells. Telophase में nuclear membrane envelope is formed. Nucleolus is formed. Chromosomes will undergo decondensation. On decondensation, again from chromosomes, chromatin is formed. Nucleolus reappears. Nuclear envelope will again form. Cell continues to elongate. So this is what happens in telophase. अभी हमें देखेंगे plant and animal cells in cytokinesis. Cytokinesis में parent cell will divide and form daughter cells. Okay, they divide and form daughter cells. Animal cells में furrow method, which is formed by furrow method. Plant cells में which is formed by the plate method, the cell plate method. Whether it is animal or plant, finally एक parent cell से दो daughter cells आते हैं. After completion of cell cycle, from one parent cell, two daughter cells are formed with same chromosome number. इसमें chromosome number same रहता हो. Whatever the chromosome number present in parent cell, वही chromosome number daughter cell में रहता. Okay. And the parent cell and the daughter cells are identical. There are no changes. Identical रहता. Okay. So mitosis takes place in most of during growth and development. Generally, mitosis is useful in growth and development. It helps in to increase number of cells. Growth and development में होता है. And healing of wounds. If you get any injury, cells are damaged. New cells are formed by this cell cycle. Healing of wounds. It is helpful for regeneration, healing of wounds, growth and development. Both are applications in cell cycle in the life of living organisms. Okay. So here we may get questions based on this. That is, from one daughter cell, one parent cell, we get two daughter cells. और इसमें chromosome number same रहता. So you should remember, for solving these numerical questions based on that is cell cycle, the logic you should remember is from one parent cell, two daughter cells are formed. और chromosomes में same number of chromosomes रहता. daughter cell और parent cell में chromosome number same रहता. For example, if there are 46 chromosomes in parent cell, daughter cells में कितने chromosomes रहता? 46. Daughter cells में भी 46 chromosomes रहते, and they are identical. Okay. From one parent cell, how many daughter cells are formed? 2. Okay. So you should remember these two logical things to solve numerical questions in NEET exam. NEET exam में इसमें ए पॉइंट्स बेस करके क्वेश्चंस आने की संभावना है. इस पॉइंट, that is, here if we take from one cell, two cells are formed, that is double the number of parent cell. It becomes double. Okay. So here you should be able to calculate how many daughter cells are formed, how many mitotic divisions will take place. Okay. So based on this logic, you should solve these numerical questions. Okay. So here comes the point, the diploid and haploid. What is this diploid and haploid? Haploid means single set of chromosomes, the single basic set of chromosomes. Diploid means two sets of chromosomes. Diploid means two sets of chromosomes. If you take, for example, humans, human diploid number is 46. Haploid number is 23. So where this diploid and where this haploid is present? Haploid is present only in gametes. सिर्फ gametes में रहता, male और female gametes में रहता. That is, सिर्फ gametes में haploid condition रहता. ये पूरा cells में whatever cell you take, इसमें diploid रहता. इसमें क्या रहता है कि diploid. Okay. Like in the plant also, if you take except male and female gamete, all cells are diploid. And when you come to particularly plants में triploid condition भी देख सकते. ये triploid condition किधर रहता? Endosperm में रहता. 3 sets of chromosomes. 3 sets of chromosomes. Okay. So in cell cycle, from a diploid cell, a diploid, 2 diploid cells are formed. Gametes are formed by meiosis. There is next cell division process, that is meiosis, by which male and female gametes are formed. Okay. So here you should remember the terms, what is meant by diploid, what is meant by haploid, what is meant by triploid. Triploid किधर रहता, haploid किधर रहता, diploid किधर रहता. Based on this also, we will get questions. Based on this, we will get need-based questions. Diploid, haploid, triploid, you should remember. Okay. So diploid means 2 sets of chromosomes. So like this, you should remember these basic things to solve numerical questions. To solve numerical questions. Just for example, if you take one bacterial cell undergoes. Okay. Cell cycle for every 20 minutes. Remember the question carefully. One bacterial cell undergoes cell cycle for every 20 minutes. So after 40 minutes, how many daughter cells are formed? Okay. Just take one. Pella 20 minutes. 2 or 4. From these 2 after 20 minutes, 4 or 4. From each one to totally 4. That is, after 40 minutes, we will get 4 daughter cells. After 40 minutes, we will get 4 daughter cells. Okay. So this is how you should remember you should solve the numericals based on number of mitotic divisions. Okay. So while you are solving numericals, you should write the data. Whatever the data is given, it should be written in a proper format. Here the data is for every 20 minutes, one cell cycle takes place. One cell undergoes cell cycle for every 20 minutes. This is the thing you should write. One cell. Based on this, you can solve such type of questions. Okay. So like this, if we take for next, if we take 80 minutes, the cell number will become 8. Every time it becomes double, like next 20 minutes it will become 8, next 20 minutes 16, next 20 minutes 32. The number of cells will become double at every 20 minutes. This is a sample neat question based on the cell cycle. Okay. And here you should remember what is meant by diploid, haploid, triploid. where triploid condition is seen, where haploid condition is seen, where diploid is. So if you remember the haploid, where we'll get haploid like male and female gametes. Triploid, where in plants you'll get, it is seen in endosperm, and remaining all are diploid of leaf cell, lelu, stem cell, root cell, and meristem cell. Any cell you take, it will have diploid condition, except male and female gametes and endosperm. And you should remember in the life cycle of plants, where what are diploids, where will get diploids. Like for example, if you take pollen grains haploid rectum, male gametes haploid rectum, egg cell haploid rectum, synergids haploid rectum, antipodals haploid rectum, polar nuclei haploid rectum. पूरा plant life cycle, angiosperm life cycle में cells इसका प्लॉइड और एंडोस्पर्म ट्रिप्लॉइड रहता, बाकी पूरा सेल्स डिप्लॉइड रहता। ऐसे हमें एनिमल्स में कोई एंडोस्पर्म नहीं रहता, सिर्फ मेल और फीमेल गैमेट्स। Okay, so here if we take in animals like secondary spermatocyte, spermatids, sperms, haploid rectum. Okay, like secondary oocyte, polar nuclei, ovum, haploid rectum, pura baki, diploid rectum. So this is how you should remember which is diploid, which is haploid, which is triploid. And while you are studying life cycle, we'll come across many stages like this, like when you are studying the reproduction part specifically. For example, if you take Spirogyra, the plant body of Spirogyra itself is a haploid. Okay, itself is a haploid. There is no need of mito, there is no need of meiosis there. Okay. These are some exceptions you should remember. Generally, the plant body will be diploid, but in some organisms it will be haploid, like Spirogyra, which shows haplontic life cycle. So you should remember these exceptions and these numericals to solve the questions based on cell cycle. Okay, students, thank you all. We'll meet in the next session. Follow us SPSVERSITY on social media.